Hello, Assalamualaikum Abi to everyone who is watching, especially my panels. Welcome to my final year presentation entitled Preliminary Study of Bats Diversity and Distribution in UiTM Pahang, Jengka Campus. But before that, my name is Alia Zunairah Mohd Azlan with my metric number 2019612912. Throughout this study, I am supervised by Sir Muhammad Azam Akmal bin Abu Bakar. When we are mentioning about bats, we must be reminded of coronavirus. Most people are scared of these nighttime flyers, probably because of their dark colors and some out and about rumors about them. However, this creature is actually a gentle one, especially to a forest. Bats belong to the order of Chiroptera and is divided into two suborders, Megachiroptera and Microchiroptera. They are the only flying mammals that are capable of true flight. They are significant in forest preservation as they are an excellent seed dispersal. So, what do you understand about that statement? In easy words, when they are feeding on fruits, they only suck the juices out of the fruit in order to gain the nutrients needed. After that, they usually throw it on the ground, hence new plants. Other than consuming fruits, other species feeds on pollens, nectar, leaves, insects, and other small mammals or even blood. They either live solitarily or colonies. The Chiroptera are well diverse and can be found in all continents except for Antarctica, South America, North Scandinavia, and a few isolated islands. Moving on to the problem statements. First, small mammals fauna in UiTM Jengka is poorly documented. Hence, the needs to gather information on the species composition and species diversity in UiTM Jengka is crucial. The objectives of this study are to identify bat species in UiTM Jengka and to find out the diversity richness of bats in UiTM Jengka. There are a few steps when it comes to experimenting. First, selecting my study sites. My chosen study sites are in the residency area of College Tuk Gajah, which is Zone A, and another one is at the Sri Gadings Forest Range, which is Zone B. Second step is the bat sampling. The sampling was done by installing mist nets from 6 p.m. to 7:30 a.m. The sampling was done in 10 appropriate days in each zones. Third step is a bat's identification. Bats were untangled and was placed in a soft cloth bag. Soft cloth bag was chosen because of its breathable material. Next, we measure the external features and weigh using the electronic weighing scale. Then, the bats were marked and released. For identification, trustable sources were used. Final step is the calculation of diversity index. The whole calculation was generated using the paleontological statistics software, which is known as PAST. It calculated Shannon-Weiner index as well as Simpson's diversity index. On to the most exciting part, which is the results. In this study, one family, 3 genuses, 6 species, 29 individuals were recorded in total. All species are from Theropodidae family, the frugivorous bats. In Cynopterus genus, there were 4 species. In Pentatha genus, 1 species, and Rosetus, 1 species. Out of all 29 individuals, 5 were found dead, and 1 of the deceased individuals were unable to be identified. These are the 4 Cynopterus species that were recorded. There are Cynopterus burkhiatis forest, Cynopterus burkhiatis sunda, Cynopterus sphinx, and Cynopterus horsfieldi. These species are arranged accordingly to the body size. Means Cynopterus burkhiatis forest has the smallest body size compared to the others. They all have similar features and colorations. Orangey color with white-edged ears and wing bones. According to the studies, forest and sunda has a very similar features, but it can be distinguished by their habitats and their forearm length. Next is the genus Pentator. This is Pentator lucasi. It has dark grey-brown upperparts, pale buffy grey underparts, darker head, and dark edges eyes. The last one is the genus Rosetus. This is Rosetus leschenolti. This species is the only cod species that has elongated muzzles. It has grey-brown to buffy brown upperparts with paler underparts. The fur is short and sparse except for the chin and neck. This genus is the only echolocating fruit bats. Other than this genus, the other Theropodidae are using their eyes to navigate. This is Zone A. Zone A is rather an urbanized area near the College Tok Gajah and Sri Gading Forest, where the net was installed under a shaded area of understory plants. It is also surrounded with other forms of nature, such as a small man-made lake, along with the potentially bats' food sources like fruit trees and other flowering plants. Based on the chart shown, Zone A acquired 18 individuals representing 5 species throughout the 10 days of sampling. They are C. Berchitis forest, C. Berchitis sunda, C. horsebills, C. finks, and Pilucasi. As for Zone B is a simpler and much more isolated area which has lesser disturbance and better ambience. The mesh net was installed under thick trees or known as Pokok Jati or Tectona grandis and other shrubs. Tectona grandis is a type of well-known timber tree with large, simple leaves. 11 species representing 5 species were caught, which are C. Berchitis sunda, C. horsebill, C. finks, Pilucasi, and Rosetus lachenolti. However, 5 individuals are dead and 1 is unidentified. The diversity index have been calculated using past software. Shannon-Weiner index shows that Zone A has higher index compared to Zone B. Meanwhile, for Simpson index, Zone A has greater value compared to Zone B. This also makes the evenness index higher in Zone A. Abundancy and diversity of the species that was proven in the data are relatively consistent. Hence, we can say that as the number of species increase, the diversity index value also increase. To conclude, Cynopterus finks dominated the data with 9 individuals. Cynopterus genus has the most number in collected data. Diversity index in Zone A has dominated the results, which consist of high species richness as well as evenness. Zone A is more diverse and even compared to Zone B. As for recommendation, data collection such as traditionally recognized by morphological characteristic can be improved by using other advanced methods. Hence, more identification by genetics such as molecular study like DNA barcoding should be done in the future. As for study of distribution, future studies should take into account the use of Geographical Information System, which is GIS, which creates database and maps to acquire highly accurate distribution of Chiroptera. That is all from me. Thank you.